be doing the role of the pancreas and liver in regulating digestion. We'll talk about the physiological functions of the liver and the pancreas. The function of the liver, the carbohydrate metabolism, where stored glycogen and gluconeogenesis occurs. That's where glucose is taken in the form using glucagon and stored in the liver for energy or for rest and fat metabolism. Fatty acid is metabolized in acetyl coenzyme A. It enters into the citric cycle and is converted into ATP. Excess acetyl coenzyme A is converted into acetoacetic acid, which is now taken up by the bloodstream and then converted back to acetyl coenzyme A for metabolizing. The liver is one of the largest organs. Here I have a picture from Netter, both the lobes of the liver, the bile duct, the falciform ligament. You can see both the posterior and the anterior view of it. Blood entering the liver brings nutrients and foreign substances from the digestive tract. Altogether, bilirubin from the hemoglobin breakdown and the metabolites from peripheral tissues in the body. In turn, the liver excretes some of these in the bile and stores or metabolizes others. Here you can see how the liver functions in hormone in three different zones where the blood is in the liver, and how toxins, medications, and everything goes through the arteries and the veins and the hepatic portal system. You can see the synthesizing and the proteins and the different things that go on in our liver. It's one of the most important functioning organs in our body. The physiological functions of the liver is for protein metabolism, synthesis of plasma proteins, mostly albumin. And it's important for the synthesis of drugs and hormones, and to bring osmotic balance. We'll discuss that more later when we talk about bile. The active albumin causes imbalances in the Starling forces, contributing to edema, imbalance to free-bound and bound hormones and drugs. Again, like our liver is one of the main functions is to store iron, and it does that in the form of ferritin. Also, it deanimates amino acids, meaning it takes away like a protein chain from the amino acid, and it turns it into urea and ammonia. It's also important for storage of vitamins such as vitamin A, D, and B12. It's very important for our bodies. And the physiological functions of the liver, we can see the gastrin, the hormones, secretin, and how it stimulates, acids, and the parenchymal secretion. That's just a mixture that constantly flows in our digestive tract. So you see the entering of that and the bile storage. Sphincter of Oddi, usually that's very significant when you are looking at the gallbladder, and a lot of times if there's like gallstones, people will get their gallbladder removed, and usually that is the sign of where the gallbladder is beginning, the sphincter of Oddi. So the detoxification of endogenous compounds, hormones, and drugs. The Cooper cells are residentially like macrophages that clear out toxins inside the gut, as I discussed earlier in zone 1 of the liver. There's 3 different zones, but the highest concentration of oxygen and synthesis of drugs and toxins are done in zone 1. The physiological functions of the liver. The production of bile in the liver and stored in the gallbladder. It's secreted from hepatocytes and made up of water, salts, cholesterol, and electrolytes. Actively transports sodium to create osmotic imbalance. Digestion, bile emulsifies fat and fat-soluble vitamins like vitamin K and D. Bile salts, hepatic metabolism, and bilirubin are also major parts of the synthesis occurring in the liver during digestion. Bile salts, fats include 90% of triglycerides, cholesterol, phospholipids, long fatty acid chains, and fat-soluble vitamins. I kind of took a picture from Netter again, showing the vein system. There's also the artery, but you can see the main superior mesenteric vein that goes to the side of the body, and the inferior mesenteric vein. Superior being more significant because it takes all the nutrients and everything we need into our large intestine. Um, you can see how the blood flows out, the hepatic portal system, and also the accessory organs that we'll discuss. You see that bile is a coated lipid droplet. This is the production and absorption of fats in our digestive system, how it's emulsified, and how the environment is spread throughout. Micelles and small discs with bile salts, phospholipids, fatty acids, cholesterol, and mono and triglycerides are here, both in the hydrophobic, which is mostly what the bile salt is, since it's fat and a mixture of other things. It's considered hydrophobic. Now we'll talk about the physiological functions of the pancreas. Its two main components are the exocrine and endocrine functions. The exocrine releasing of digestive enzymes and bicarbonate ions into the duodenum. Acinar cells secrete digestive enzymes to travel through the ducts that are lined with the epithelial cells. Epithelial cells lines the duct of the exocrine component of the pancreas, which secretes bicarbonate ions, which allows for pH activation of the pancreatic digestive enzymes. Endocrine functions releases substances right directly into the bloodstream. The islets of Langerhans are heat cells that represent endocrine function of the pancreas. It secretes hormones, insulin, and glucagon, which play a crucial role in glucose, fat, and protein homeostasis. Here I have two pictures from Netter of the accessory organ as the pancreas, the head of the pancreas, and the arteries functioning around as the accessory organs. You see the gallbladder where bile is stored, and the pancreas is usually made up of some of the foods that we eat, kind of. So at the end of the day, it's really true. You are what you eat. Pancreas being one of those organs that are made up of the same substances that we find in our food. You see how the hormones and the surrounding things that take place in our digestive system, the enterocytes, the nerves, everything in our gut is part of the health and well-being of it. You see the superior mesenteric artery, inferior of the pancreas. Again, a lot of things going around it, and very important part of our bodily functions. Okay, sorry about that. The functions of the pancreas: the exocrine acini cells secrete amylase, lipase, and protease, which is important for our gut health. Amylase breaking down carbohydrates and into smaller products. Lipase breaking down lipids and carrying out fat digestion. You see the end result of one monoglyceride and two free fatty acids. So for every one, you see a breakdown of two. And the proteinase breaking down protein, trypsinogen into trypsin. This happens by alkaline pH balance of the gut via the enterokinase, chymotrypsinogen breaking down into chymotrypsin. And these are all baselines for also building, carrying out hormone regulation and forming neurotransmitters and hormones in our body, such as melatonin, which is formed from tryptophan. And also, here we talk about the anatomy of the exocrine and endocrine pancreas. The exocrine pancreas secretes digestive enzymes and sodium bicarbonate. That's a really big deal if you're thinking of the enterocytes that keep electrolytes in our gut and also pH balance. The islet cells also appear where the acini is and the duct cells that secrete and enter the digestive tract. We can see that. I think it appeared on our exam too, so this is a very familiar picture. Also, the activation of the pancreatic zymogens. Here we see how what happens after the substances enter our digestive system. The activated enzymes that we discussed earlier, and also how the bicarbonate secretion occurs. And you see sodium exiting in the ATP molecule. This is the main component. You see the bicarbonate also exiting, keeping that balance. Cells that produce bicarbonate have high concentration of carbonic anhydrase, and chlorine enters the cell indirectly and active transport and leaves the apical side through the CFTR channel. Leaky junctions allow paracellular movement of ions and water and negative ions in the lumen that attract sodium by the paracellular pathway. So we can say that our stomach is very chlorinated, I guess, and like there's a lot of surrounding fluids, interstitial fluid, and pancreas is very much a big part of when it comes to balance. Here's another image of carbohydrates breakdown of monosaccharides, which is done through amylase. Also over here we have the breakdown of proteins through protease and peptide absorption after digestion. Proteins are absorbed mostly by free amino acids. The tripeptides are absorbed. Some peptides are larger, and tripeptides can be absorbed by transcytosis. So that is the end of my slideshow. Thank you for your time, and I hope you enjoyed it.